I'm Melissa Harrington. I'm currently the Associate Vice President for Research at Delaware State University and the PI of the Delaware Center for Neuroscience Research, a COBRE grant that links University of Delaware and Delaware State University. And we are just finishing our second year of Phase 2 awards. So we've been going for a while. I guess we're saying we're really hitting our stride now. We have two core facilities that are supported partially by our COBRE grants that are open to researchers across the state of Delaware and potentially beyond if anyone wants to use them. And so I wanted to share a little bit about them because one is brand new that's based at Delaware State University that not many people might know about. And we have just started a cell electrophysiology core here at the university. And this is a unique research resource in the state. That's not something that anybody else has. And in fact, somewhat to my frustration when I first came here as an early stage investigator and was trying to get help getting my research going, there are very few electrophysiologists in Delaware, period. So we think that creates a potential research need that our core facility can fill. So if you have the screen sharing, if you're called in, what you see, there's a picture of a few of the guys who work in the center. So right now we have two research scientists, faculty or sort of research faculty at Del State that are part of our Delaware Institute for Science and Technology that are the primary staff of the core. So Julian, if you have the screen share, that's Julian Wiltorton. He's the white guy. And then Jean-Lee Sun is the Asian guy in the center of the picture. And they both contribute half-time to the core. And then also in the picture is one of our new graduate students who is learning how to do LTP recordings in hippocampal and hippocampal and cortical brain slices, which is something that a lot of people in the neuroscience field might be interested in as an assay for function in a mutant or a transgenic animal. So what the cell electrophysiology core does is to characterize function in excitable cells and tissues, brain slices, and then also in ion channels. So the next slide in the PowerPoint. So the services we offer, one of them is traditional patch clamp. This is one cell at a time. I have some very nice images of neurons being recorded and some data. So with traditional patch clamp, we can record whole cell currents from cells that are cultured or in tissue slices that give a picture of the overall ionic or electrophysiological function of neurons, or can look at particular currents. And then it's also possible to do, and those are, if you're seeing the screen, those are the top two images. It's also possible to record single channel activity for a detailed study of ion channel function, and that's in the lower part of the screen. There's some beautiful images of opening and closing ion channels and the current voltage relationship. We also offer multi-electrode recording with a new MultiChannel Systems apparatus. So shout out to Embry. We purchased this with Embry carryover funds last year. And what a multi-channel system allows, this is the most efficient way to record LTP because you don't have to find, you have, in our case, 64 chances to find the pairs of neurons that are connected. If you have one or two that are stimulating, then you have 62 chances to find the neurons that are receiving that stimulation or that are postsynaptic to the cells you're stimulating. And you can also get a picture of the global activity across a tissue slice or a culture of a primary culture from a transgenic animal or induced pluripotent stem cells if one wants to show that they are actually acting like neurons instead of just expressing neuronal markers. A multi-electrode recording system of the cultured, using multi-electrode recording system for the cultured cells is the best way to do that. We also, brand new, have an automated patch clamp system. It's the Ion Flux Mercury. Right now, all we could afford was the 16-channel system, but we're hoping if we get a little more money to be able to upgrade that to the 64-channel system. And this is a really efficient way to measure for patch clamp, it's what patch clampers would consider a high throughput way to measure ion channel function. So the way the automated patch clamp systems work is your channel is expressed in a cell line. We're using CHO cells right now, but they are then grown in culture and they have to be suspended. So the ion channel Mercury, the 16 that we have, allows you to record 16 cells in parallel, and it's not going to give single channel currents like traditional patch clamp does, but it shows the overall current through a class of channels. You know, you can silent. The nice thing about CHO cells is they're quite silent on their native ion channels. They have very few native ion channels, and so you can either stably or transiently express a mutated channel in that cell line and be able to measure the current. The other thing we're working on right now is a way to do this in a culture or in a primary culture. So the key for these automated patch clamp systems to work, the cells have to be greater than 90% homogeneous. So you really need all the same cells, and you need a lot of cells. That's why traditionally people are doing these in cell lines. We're working to try and get this to work in cerebellar granule cells, in primary, isolated cerebellar granule cells. They might be the one uniquely suitable cell in a real nervous system because they are so numerous in the cerebellum, and I mean they're almost half the neurons in our brain are cerebellar granule cells. They're so numerous, and it's possible to isolate them to greater than 90% purity relatively easily. And the idea if we can get this to work in cerebellar granule cells is that if people want to see their ion channel mutation in a native mammalian cell with the native regulatory structure, say all the beta subunits and all the native regulatory structure of the neuron, then they could express that channel in cerebellar granule cells, which is relatively easy to do. There are Cree markers, there are Cree lines out there to do that. And then by isolating those neurons and recording them in the automated patch clamp system, you could get a idea of the overall, you could get a sort of high throughput measure of the function of the channels. Okay, so summary, the electrophysiology services that we're offering. So primary cell cultures, cell lines, acute slices, cultured slices. For the animal work, our animal facility, our mouse facility is very small, so we can't really do the husbandry here. For people that are interested in slice recording an acute slice recording from animals or from primary cultures, it would require bringing the animals to the lab. Fortunately, it's a terminal experiment, so we would be able to get a protocol approved where the animals remain in the lab for a day or two until they're sacrificed to use to collect the data. So it should be possible to do acute slice work pretty easily. So experiment types, so we can do basic cell membrane properties, action potentials, ion channel currents, look at synaptic transmission, synaptic plasticity. The setup we have for the traditional patch clamp combines fluorescence imaging as well. So it would be possible to do sort of correlate the calcium imaging along with the patch clamp recordings. There's also the multi-electrode recording, And we also have, again, in our traditional patch clamp set up for optogenetics stimulation as well. Just to give you an idea of the fee structure, which this isn't complete, but I couldn't find the most recent in the 10 or 15 minutes this morning I gave myself to put this together. I couldn't find the most complete description. So basically, the prices are really reasonable. So for trained students or researchers that just want to use the equipment, particularly for the multi-electrode array equipment, that's a really easy training. The science of multi-electrode recording is mostly in the analysis, not in the data collection. So that's only $10 a day just to access the machine to record the system. We're going to be charging only $10 a day, while the training's about $8 an hour. Traditional patch clamp, these are the most time-consuming ones. We're charging $100 a day per, or $100 per experiment day, and so Jean-Lee and Julian both have more than a decade of experience in traditional patch clamp, and so they're able to do about 15 to 16 cells a day. So you can get a lot of data in a day. So that's the basic experiment. Some things that if you're doing, it's $100 a day for cell recording from cells, because those are much faster, and then $150 a day if it includes slices, which is where a significant part of the time is preparing the slices, but still to get the same amount of data analysis. It also includes preparing, not only collecting the data, but doing the analysis and preparing the figure, because we're trying to make electrophysiology studies accessible to other kinds of researchers who don't have the expertise or the interest in developing the expertise to do the analysis. Field potentials, like I say, just accessing the equipment is only $10 a day, but if Julian or Jean-Lee or one of our students is going to be actually collecting the extracellular field potentials, say for a long-term potentiation experiment, that will also be about $100 a day. They for just consultation and advice, right? That's free about electrophysiology. The automated patch clamp, we're still kind of settling on what we're going to charge. The cassettes that collect the data and are about $350 each. The cassettes that the machine uses for collecting the data are about $350 each. So that's basically, and it can take, about half a day to do the experiment that will use one cassette. They are not reusable. So we're talking potentially just in the machine just in the supplies for the machine, about $750 a day. So that's going to be fairly expensive. On the other hand, the amount of data you get from that can potentially be nearly the complete experiment because of the parallel nature of it. So there are, and the way this would work in general is that because of the kind of level of expertise that's required, this is not simply like coming in to use a microscope. People that are interested in using the core, we would establish a collaboration. And it would not be fee for service, but rather a collaboration, and the fee would be kind of to offset the time and the material supplies. Let's see. So that's the cell electrophysiology core. I should say the other, it's at Delaware State University, University of Delaware. The COBRI partially supports the Center for Brain and Biomedical Imaging, which some of you might be, probably most of you are familiar with. That has, for a few years, since the beginning, has had a Siemens 3 Tesla magneton prisma scanner, which is used for human MRI and fMRI and all kinds of magnetic resonance imaging of all kinds. That is my understanding. The cost for that is about $400 an hour, or is $400 an hour. Just coming online, still being tested, but I think going to be available for general use very soon, is a small bore 9.4 Tesla Bruker MRI scanner that is for small animals. And the charge for that is going to be $200 an hour. And I should say there is pilot funding available to offset those costs through the COBRI grant. And that funding is available through the Center for Biomedical and Brain Imaging. So there are pilot funding applications on the website. There's a pilot funding application that you can use if you are a first-time user and want to collect some data to support a grant proposal or to test an idea. There are also INBRI Core Access Awards that will pay both for the electrophysiology services and for the MRI services through the CBBI, and that you would apply when a researcher would apply through the INBRI site. And that's what I had to say about the core facilities that we're supporting with our neuroscience, Delaware Center for Neuroscience Research. I'll answer any questions to the best I can.
Thank you very much.